In normal tissues, the rate of new cell growth is balanced by old cell death. New cells are growing, old cells are dying. So that comes in a proper balance. The moment the balance is disrupted, this process begins. But there are many, not just one factor, but multiple factors behind this change. This disruption can result from uncontrolled cell growth and loss of cells' ability to undergo apoptosis. You know what is apoptosis? That is programmed cell death. That is the normal cell programming. If that is disrupted, the cell death does not happen, so there is one more accumulation. What are the causes of cancer? Various factors, and each of them are separate questions. Like carcinogenic causes, chemicals, that is chemical carcinogenesis, radiation carcinogenesis, some virus, that is infective factor, and heredity. So we will now discuss them in detail. So what is the molecular basis of cancer? The molecular basis of cancer, it is a genetic disease, mutations in the genes which results in alteration of proteins during cell division, during the effect of external agents, or sometimes pure bad luck, that means random event. Most cancers result from mutations in somatic cells. Some cancers are caused by mutations in germline cells. Somatic cells are most commonly mutated in the cancer. I will always point out the important points for viva and theory. Keep a note of that, those things. Onegula can list such important lists. So cancer is multi-step process. What is that? Carcinogenesis is a multi-step process resulting from accumulation of multiple mutations. Just single mutation will not cause cancer. Accumulation of multiple mutations, and there is a thing called Knudsen's two-hit hypothesis. Knudsen's two-hit hypothesis. Just write down the name, Knudsen's two-hit hypothesis. Read it sometime later for your understanding. Knudsen's two hits are needed. Just one mutations or one hit may not cause cancer. Two-hit, Knudsen's, K-N-U-D-S-O-N-S, Knudsen's two-hit hypothesis. So, accumulation of multiple mutations. These mutations accumulate independently in different clonal cells, generating subclones. Over a period of time, tumors not only increase in size but becomes more and more aggressive. With time, it becomes more and more aggressive, as we know from the history of many patients. Initially, that cancer is growing slowly, then it becomes more rapid. And even more in case of metastatic cancers. This is called tumor progression. Tumor progression means rate or aggressiveness is increasing with time. This is the point you must know. 7 points you must realize and retain in your mind for viva especially. I will at least ask these 7 points. What are the 7 main alterations in cancer? Main alterations in cancer, this is the basic mechanisms. Just write down the points. 1. Self-sufficiency in growth signals. Self-sufficiency in growth signals. Points gulo mone rakhte hobe. Yaad rakhna hai ye sab points ko. Tumors have, what is that? Tumors have the capacity to proliferate without any external stimuli. Usually, normally the cells will proliferate only when there is external stimuli. But in case of cancer, without even external stimuli, the cells will proliferate on its own, keep on proliferating. So this is self-sufficiency. It is self-sufficient, does not need external factors. Self-sufficiency in growth signals. Number 2. Insensitivity to growth inhibitory signals. There are various mechanisms where the excessive growth can be stopped by some inhibitory factors or inhibitory proteins. If these inhibitory factors themselves are inhibited, so that will lead to more and more progression of cancers. Inhibition of the growth inhibitory signals. The things which inhibit themselves are inhibited. Tumors may not respond to molecules that are inhibitory to the proliferation of normal cells, such as 2 names, TGF-beta, transforming growth factor beta, and direct inhibitors of cyclin-dependent kinases, CDK1. So first point was self-sufficiency in growth signals. Second point is insensitivity to growth inhibitory signals. Third point is evasion of apoptosis, which we were already discussing. The apoptosis, the normal mechanism of cell death, is evaded by these cells. So tumors may be resistant to programmed cell death. As a consequence of inactivation of P53. Very important. Inactivation of P53. This we will discuss later separately, and this will also come as a short note. P53. And also extremely important in MCQ sections. Inactivation of P53 leads to evasion of apoptosis. Inactivation of P53 leads to evasion of apoptosis. Number 4 point is limitless replicative potential. What is that? Tumor cells have unrestricted proliferative capacity, avoiding cellular senescence and mitotic catastrophes. So, this will keep replicating. Limitless replication. 5th point, sustained angiogenesis. So, what is angiogenesis? For the growth of the cells, what we need? How will a cell grow? With the help of nutrition. So nutritions come in the form of blood, yes. Blood, that is vascularity. The blood will give the nutritions, oxygens, exactly. So this is called angiogenesis. Angiogenesis is formation of blood. See here, tumor cells like normal cells are not able to grow without formation of a vascular supply to bring nutrients and oxygen and remove waste products. Hence, tumor must induce angiogenesis. Tumor cells themselves have separate ability to perform angiogenesis. তারা নিজেরাই নিজেদের সেলের সমস্ত কিছু ব্যবস্থা করে নিচ্ছে, কোনোভাবেই তাকে আটকানো যাচ্ছে না, ধ্বংস করা যাচ্ছে না, and the nutrition is they are funding themselves. So it is very difficult to kill these cells. Ability to invade and metastasize. Invasion is a very important point of cancer spread, as you know. Tumor metastases are the cause of vast majority of cancer deaths and depend upon processes that are intrinsic to the cell and are initiated by signals from the tissue environment. So it is causing invasion, invading to the other cells and metastasizing. Defect in DNA repair. Old cells, there is a defect in the DNA, that DNA defects are repaired. But here, that process itself is problematic. So what is it saying? Tumors may fail to repair the DNA damage caused by carcinogens or incurred during unregulated cellular proliferation. During unregulated cellular proliferation, whatever DNA damage is happening, these cells may not be able to repair them. So, have you read oxidation and those things? Oxidation, reactive oxygen species, and like heard of those things? No? Antioxidant, that is the mechanism, that is the concept. Why are we taking antioxidants? Vitamin C, vitamin E, why are we taking? These are removing the reactive oxygen species, and that is helping in the repair of the DNA. Here, this process itself is defective. So these are the 7 points you must remember. Pura ratta mana hai, ye 7 point. I will obviously ask these questions in the exams. 7 points. So now the few of the important points we will discuss in detail, not everything. What is self-sufficiency in growth signals? In a normal cell, proto-oncogenes have multiple roles. What is proto-oncogenes? That is the precursor of oncogene. Here what happens? The proto-oncogene is mutated leading to defective oncogene. So what is this defective oncogene doing? This defective oncogene is, self-sufficiency for growth to cancer cell is provided by oncogenes which are mutant proto-oncogenes. So these are mutated and becomes defective. What does it cause? Mutations convert, inducible proto-oncogenes into constitutively active oncogenes. So the oncogenes are constantly active. In normal cells, the genes are sometimes acting, sometimes they are in inhibitory mode. But here, due to the mutation, it is constantly active and always replicating. So this is called self-sufficiency in the growth signals. These are the points you must know. What are the oncogenes? Growth factor receptors, signal transduction proteins, nuclear regulatory proteins, and cell cycle regulators. This is a very high yield MCQ topic, a very high yield MCQ zone even for NEET PG. Because fundamental টা strong না হলে এখানে ভীষণ গুলিয়ে যায়, very close options they are মানে always given, তো খুব পরীক্ষার সময় প্রবলেম হয়. What are the growth factors? Growth factors, normal cells require stimulation by growth factors to undergo proliferation. Normal cells proliferate by the effect of growth factors. Mostly these growth factors are secreted by one cell type and acts on a neighboring cell to stimulate proliferation. What is this? Cancer cells acquire the ability to synthesize their own growth factors. own growth, যেটা একটু আগে আমরা বলছিলাম, সেলফ সাফিসিয়েন্ট হয়ে যাচ্ছে, বাইরের সেল থেকে লাগছে না। নর্মালি কি হচ্ছে, পার্শ্ববর্তী পাশের সেল থেকে growth factor টা আসছে। Some neighboring cells are secreting the growth factors, but here in case of cancer, the cells themselves are procuring themselves. So what are the examples? Glioblastoma secrete PDGF. Remember these two? Glioblastoma secretes PDGF. Write down. Glioblastomas secrete PDGF. Sarcoma secrete TGF alpha. Two examples is enough. Glioblastoma secrete PDGF and sarcoma secrete TGF alpha. So, where will the growth factors work? In the growth factor receptors. No? Several oncogenes that encode growth factor receptors have been found. Like transmembrane proteins. Physiology में ये सब पढ़ा है ना तुम लोगों ने? Growth factor receptors and transmembrane proteins. Transmembrane proteins with an external ligand binding domain and the cytoplasmic tyrosine kinase domain. Ligand binding domain, tyrosine kinase domain. Two domains you have to remember. In normal forms, the kinase is transiently activated. In normal cells, this kinase is transiently activated, but in oncogenic versions, kinase is constitutively activated, resulting in continuous mitogenic signals to the cell, even in the absence of the growth factors. This is the understanding of the topics that we were discussing before, how this self-sufficiency is working. Normally, this kinase is only transiently activated. Kinase. See, this cytoplasmic tyrosine kinase domain, the growth factor is coming from the outside cell and it is working on the cell receptor, which is tyrosine kinase. This tyrosine kinase normally is only transiently activated. Only when the external cells are secreting the growth factor, then it's only working. But in case of cancer cells, what is happening? Kinase is constitutively activated, resulting in continuous mitotic signals, even in the absence of growth factor in environment. So here the neighboring cells are not secreting growth factor, but it is on its own constitutively activated. So this previous point, this growth factor is working in this because of constitutively activated tyrosine kinase. Constitutively activated tyrosine kinase. Remember this point? Examples. Please take down the notes. এবার পরপর অনেকগুলো লিস্ট আসবে, those are very very high exam points. This is a bit difficult topic, so I am trying to do it as easily as possible. Now today I cannot say that you should read today, but after the fest is over, please go through this topic. So, rate dominantly inherited main types, rate leading to, see what is the list saying? Examples of growth factor receptor oncogenes and associated cancer. So rate is associated with main type 2A, 2B and familial medullary thyroid carcinoma. So you can obviously understand this is a very important area for viva. সব কিছু গুলিয়ে যায় কিন্তু পরীক্ষার সময় এগুলো। Rate leading to main 2A, 2B and familial medullary thyroid carcinoma. C-KIT mutation leading to gastrointestinal stromal tumors. ERBB1 and ERBB2 very important. You have to write, draw pictures also during the exams when these questions come, how the mechanism works, oncogenesis. ERBB1 leading to squamous cell carcinoma of lung, glioblastoma, head and neck tumors. ERBB2 also known as hard-to-new. Hard-to-new you have already heard in the breast carcinoma, I think. See, breast cancers and in human adenocarcinomas arising within the ovary, lung, stomach and salivary glands. So list ho gaya. Next slide we can go. Nuclear regulatory proteins or transcription factors. Transcription factors. What are these example? MYC. Very, very important. MYC is a nuclear regulatory protein. What is transcription factors? MYC is a nuclear regulatory protein with a broad range of activity which includes mechanism গুলো মনে রাখতে হবে কি আছে what are the mechanisms of action of MYC? Histone acetylation. Histone acetylation 1. 2 is reduced cell adhesion. 3 is increased telomerase activity. এই তিনটা at least মনে রাখতে হবে. Important role in selection of origins of replication. Overexpression of MYC may drive activation of more origins, so multi-origin expression. So this bypasses the checkpoints involved in replication. Checkpoints involved in replication leading to genomic damage and accumulation of mutations. So MYC is a regulatory protein. The activation leading to bypass checkpasses involved in the replication. এর মধ্যেই পয়েন্টস গুলো রয়েছে, just you have to write down the basic points. Bypass the checkpoints involved in the replication leading to accumulation of mutations. So what is the example? Dysregulation of MYC seen in Burkitt's lymphoma. এগুলো কিন্তু তুমি যখন তখন ধরবো আবার। MYC dysregulation হচ্ছে Burkitt's lymphoma. MYC is amplified in breast, colon, lung and other carcinomas. তাহলে amplification and dysregulation. Amplification হচ্ছে breast, colon, lung CA and dysregulation হচ্ছে Burkitt's lymphoma. What are the cell cycle regulators? You know the cell cycle now already. So in the various checkpoints of the cell cycle, there are many regulators which are controlling the cell cycle. If there is any difficulty, that also leads to carcinogenesis. See? G1S, G2M, you know already, na? G1 stage S, G2 stage and M. In this checkpoint, see here, this one checkpoint, here is one checkpoint. G2 to M, cyclin B and CDK1. Cyclin B and CDK1 is acting. See, in various checkpoints, there is working cyclin D, CDK6 for G1 to S and G2 to M. These are the two main checkpoints. What are the two important checkpoints? G1S and G2M. Example of cell cycle regulator genes and associated cancers. Overexpression of cyclin D genes, note down the points. Overexpression of cyclin D genes, breast, esophagus, liver and subset of lymphomas. Where is cyclin D working? See here. Cyclin D working CDK4 via CDK4 and CDK6. During the G1S cycle, G1S part of the cycle, if there is a problem here, it will lead to cancer of breast, esophagus, liver and subset of lymphoma. Overexpression of cyclin D and amplification of CDK4 gene leading to melanoma, sarcoma, glioblastoma. ये पूरा रट्टा मानना ही होगा, এটা কেন কিছু করার নেই। You have to understand the basic mechanisms and remember the cancers associated. Just basic, in a simple way that I am saying. Not very complicated or not very detailed, but the basic you must be clear about that. Okay. Germline mutations in P16 leading to melanoma. See, CDK inhibitors. This CDK4, CDK6, CDK2, CDK1 is working. This P16 is inhibitor of these CDKs. If this P16 is defective, then this CDK inhibition is not working properly. See P16 there, leading to melanoma. Somatically acquired deletion of or inactivation of P16, pancreatic carcinoma, glioblastoma, esophageal cancers. As simply as possible, you can remember this. এর থেকে সিম্পল আর হতে পারে না, বাট এইটুকু মনে রাখতেই হবে। এই যে ছবিটা দেখবি, ছবির সাথে মেলাবি, ছবিটা দেখবি আর লিস্টটা পড়বি যে কোন স্টেপে হচ্ছে, মার্ক করে রাখবি, তাহলে মনে থাকবে। দেখতে পেলি P16, তাহলে এক্সাম্পল গুলো মনে রাখতে হবে। সাইক্লিন ডি, CDK4, P16 কোনটা থেকে কি হচ্ছে বলে দিলাম। This is just for understanding what are the what are the mechanisms? Chromosomal changes in genome of cancer cells. What are the mechanisms by which these chromosomal changes happen? Deletion, insertion, duplication, inversion, reciprocal translocation, Robertsonian translocation, ring chromosome formation, and isochromosomes. These are the mechanisms. How the nucleotides are changed during the mutation? Nucleotide deletion, nucleotide insertion, or substitution. Some nucleotides are deleted, some new nucleotides are inserted, or some nucleotides are substituted by another. Approximately 90 to 95% of all cancers are sporadic. Contrary to the common knowledge or perception that the cancers are genetic, but only 5% cancers are actually genetic. 5 to 10%. Rest are sporadic. So this is the mechanism in nutshell, what we were discussing. This will help you to remember. This slide is for you remembering and making in the copy. When you are answering the mechanism or oncogenesis, you will write like this way, very simple and less to write and very easy to remember. Proto-oncogene, which is activated, leading to cell proliferation. That we were saying, proto-oncogene to abnormal oncogene, proto-oncogene to abnormal oncogene. But in the other hand, tumor suppressor genes, which usually suppresses the tumor, is itself inhibited. Inhibition of tumor suppressor gene and activation of proto-oncogene, both leading to cell growth and proliferation. In cancers, mutated or activated oncogenes leading to malignant transformation. This upper part is very important. Proto-oncogene activation and tumor suppression genes in activation. This is what we were discussing like before. The growth factors are coming from the surrounding cells, acting on the growth factor receptors, cytoplasmic signal transduction proteins, and then to nuclear proteins and the cell growth genes. This is the pathway. This is the pathway in which the cellular proto-oncogene activation works. Secretion of growth factors, activation on growth factor receptor, then acting on cytoplasmic signal transduction, first in cytoplasm, then in the nuclear protein transcription factors, and then in the last cell growth genes. So what is oncogene? Proto-oncogene RAS is leading to oncogene mutated RAS. Always activated. That we were discussing. এখানে শুধু ছবি করে দেওয়া আছে for understanding. Proto-oncogene is mutated, leading to always activated state, always stimulating and continuous proliferation. Continuous proliferation. So this is an important slide. HRAS, what gene? Amino acid substitution and RAS family proteins. What RAS gene is affected? What kind of cancer is happening? HRAS, KRAS, NRAS. HRAS, KRAS, NRAS. Next, many important lists coming now. Proto-oncogene to oncogene, the same thing. This one is very, very important slide. Chromosomal rearrangement or translocation. Burkitt's lymphoma caused by T814, T822 and T28. Proto-oncogene is c-MYC. So I will ask, or in the MCQ will say, what is the most common translocation associated with Burkitt's lymphoma? What is the most common translocation associated with Burkitt's lymphoma? All these three options and some other options will be given. You have to write 814. I may alter the question, what is the least common? What is the least common? I will give these three options and none of the above. Then the answer will be T28. Like this, the MCQs are formed, okay? Chronic myelogenous leukemia, very important topic, separately we will discuss again. T922 in most of the cases. BCR-ABL, the names of the proto-oncogene is BCR-ABL. BCR from one side and ABL is fusion transcripted. Acute lymphocytic leukemia. So c-MYC is these are not important, these are advanced topics. Gene amplification, কোন gene amplification এ কোন cancer হয়? Another important list. This will be mostly asked in the Viva. This is for the Viva. The earlier slide was important in both Viva and MCQ. This one is only for Viva. NMYC oncogene amplification leading to neuroblastoma, retinoblastoma. I will ask what are the, give me two examples where NMYC amplification causes cancer. Then you have to reply neuroblastoma and retinoblastoma. KRAS, which is very important. KRAS, extremely important. KRAS leads to colon carcinoma and adrenocortical carcinoma. Colon carcinoma and adrenocortical carcinoma. Oncogenes are usually dominant. Gain of function mutation. There are two types of mutation, gain of function mutation and loss of function mutation. Here in case of oncogenesis, it is usually gain of function mutation which is leading to continuous and very aggressive proliferation. Cellular proto-oncogenes that have been mutated and activated. Cellular proto-oncogenes that have been captured by retroviruses. So viral carcinogenesis, when we will discuss viral carcinogenesis, there we will see this in detail. Proto-oncogenes captured by retrovirus. Retroviruses, one is HIV. Proto-oncogenes that have been captured by retroviruses and have been mutated in the process. Virus-specific genes that behave like cellular proto-oncogenes. This we will discuss during Burkitt's lymphoma and HPV-related cancers. HPV-related cancers. What is the result? Overproduction of the growth factors. Overproduction of the normal growth factors. Flooding of the cell with replication signals. Flooding of the cell with replication signals. Uncontrolled stimulation. Uncontrolled stimulation of the intermediary pathways and cell growth by elevated levels of increased levels of transcription factors. So what is the tumor suppressor genes that I was talking just 5 minutes before? That tumor suppressor genes itself is suppressed. So loss of function. Here this is loss of function. The previous one was oncogenes. Oncogenes are affected by gain of function. So leading to active proto-oncogene to very highly overactive oncogene. Whereas this one is loss of function mutation, loss of function. Its normal function is to inhibit the tumor, suppress the tumor, that is lost. So loss of function. So absence or inactivation of the inhibitor leads to cancer. Example, this slide is very important. RB gene short note debo, এইটা লিখলেই marks. RB gene, RB gene, RB gene. So what are the important points for RB gene? First you have to write RB protein controls the cell cycle in the G1 checkpoint. That we are talking G1S, that G1 checkpoint. RB protein controls the cell cycle moving past the G1 checkpoint. RB protein binds with the transcription factor E2F, E2F. RB binds with E2F, E2F. E2F is required for, what is E2F? E2F is required for synthesis of replication enzymes. Synthesis of replication enzymes. Once this E2F and RB are bound together, there are no transcription or replication. So it is inhibiting this way. Is binding with E2F and there is no transcription or replication. So active G1 CDK cyclin kinase phosphorylates RB. Normally it is controlling it. But once it is phosphorylated, see, once it is phosphorylated, it becomes activated. Phosphorylated RB cannot bind to E2F. Very, very important. Please understand it. I have made the slides as simple to understand as possible because it is a very complex topic. If you can remember this much, this will suffice. Okay. So normally RB is binding with E2F, but here the phosphorylated RB that is unable to bind with that ETF, unable to bind with that ETF. So leading to, it goes in the S phase. So G1 is checkpoint is crossed and it is going to the S phase. So disruption and deletion of RB gene, inactivation of RB protein, uncontrolled cell proliferation leading to cancer. So G1 থেকে এসে চেকপয়েন্টে G1 এ স্টপ করে দিচ্ছিল RB এই E2F এর সাথে বাইন্ড করে, যেই আরবিটা ইনঅ্যাক্টিভেটেড হয়ে গেল, ফসফোরাইলেটেড হয়ে গেল, দ্যাট ইজ দ্য ক্যান্সার মেকানিজম, তাহলে আর এই G1 এ আটকে রাখতে পারছে না, ইট ক্যানট বাইন্ড টু দ্য E2F এনিমোর, তাই তো? তাহলে এবার G1 থেকে তার পরে ফেজে এসে চলে যাচ্ছে, নর্মালি যেতে পারছিল না, নাও ইট ইজ গোয়িং leading to cancer. Please try to practice these drawings. Very simple to get marks. কম লিখে বেশি মার্কস পাওয়ার উপায়। The examiner will know that you have understood. Bas. So this was one question, RB. Another very important question, another mechanism. Phosphorylated P53 activates transcription of the P21 gene. See, this is the damaged DNA. The P53 is phosphorylated. See, P ऊपर में P53 P लिखा है, so phosphorylated. This picture is very important. Two pathways, one pathway leading to apoptosis or cell death, and another pathway leading to cell cycle arrest. P53 is phosphorylated and gets two results. One result is apoptosis or cell death, second result is cell cycle arrest. So try to remember as simply as possible. This is the simplest possible slide of P53. Phosphorylated P53 activates transcription of P21. P21, this one. P21 CDK inhibitor inhibits kinase activity, cell cycle arrested to allow the DNA to be repaired. If damage cannot be repaired, it leads to apoptosis or cell death. So disruption or deletion of P53 or inactivation of P53 protein leads to uncorrected DNA damage, uncontrolled proliferation and cancer. Understood? Very complex topic, but you have to remember as simply as possible. Otherwise, you will forget. Noted down this slide. It's a very important one. This you will not need. This slide. Please note down this slide. Tumor suppressor genes. Tumor suppressor genes. This is the gene locus. This is the function and cancer affected. Like WT1 is very important. WT1 is very important. WT1 leads to Wilms tumor, familial Wilms tumor and sporadic lung cancer. Familial Wilms tumor and sporadic lung cancer. RB that we were discussing, RB leading to naam mein hi chupa hai, RB retinoblastoma, RB retinoblastoma and small cell lung carcinoma. P53 extremely important, Lee-Frommany syndrome. We have read Lee-Frommany syndrome, na? Lee-Frommany syndrome and breast, colon and lung cancer. And BRCA1, as we already discussed, breast cancer. Very, very high yield list. This is basically cited as what is the importance of DNA repair. DNA damage, proper repair and defective repair, a proper balance is needed. Note down this one. CML, when we will discuss pathogenesis of CML. Pathogenesis of CML. We will discuss in detail this one in a separate class. But right now, note down this. See, BCR gene is on chromosome 22 and ABR is on chromosome 9. BCR ABL fusion transcription. So, translocation, transcription and translation. See, leading to BCR ABL fusion protein. BCR ABL fusion protein. Molecular basis of multi-step carcinogenesis, this is only for understanding. This kind of pictures you will get in your textbooks, either in Robbins or Harshmohan. Just go through the pictures. After reading the mechanism, go through these pictures, you will understand better that what is happening. Because पूरा लंबा पढ़ोगे तो उतना समझ में नहीं आएगा, because it's very difficult and complex topic. Just read a small part and correspond it to this kind of pictures. Then you will understand better. And immediately after completion of the topics, always do MCQ. Always do MCQ. So this is basic, you know, what is the difference between benign and malignant, everyone knows. So what is in the nutshell, what we have discussed today, the tumor progression, how the tumor progresses. This is last example of a colon cancer. This is the normal colon wall epithelial cell. Normal colon epithelial cell. Loss of tumor suppressor gene APC. Here the gene is APC. That list tumor suppressor genes, that will be APC in case of colon carcinoma. The tumor suppressor gene, which normally suppressing the tumor APC gene, lost here. So, starting off the defective process. Small benign growth or polyp in the beginning. So, see, this is the normal colon epithelial cells. Now, loss of APC, initially formation of benign polyp. Then, multi-step. See, multi-step. First step was loss of APC. প্রথমে কিন্তু ক্যান্সার হয়ে গেল না, প্রথমে বিনাইন পলিপ হলো। দেন নেক্সট স্টেপ হচ্ছে অ্যাক্টিভেশন অফ রাস অঙ্কোজিন, লস অফ টিউমার সাপ্রেসার জিন ডিসিসি। নাও it is leading to larger benign growth or edema. Then another step, see, loss of tumor suppressor gene P53 and additional mutations leading to malignant tumor. So just one mutation হয়ে গেল আর হঠাৎ করে one fine morning cancer শুরু হয়ে গেল, that is not the case. It is a very complex and multi-step process. That is what I am trying to let you understand. প্রথমেই হলো এপিসি থেকে স্মল বিনাইন গ্রোথ, তারপরে র্যাস থেকে হলো লার্জার বিনাইন গ্রোথ, তারপরে লস অফ টিউমার সাপ্রেসর জিন P53 হয়ে ফাইনালি আফটার 3 স্টেপস ইটস লিডিং টু ম্যালিগনেন্ট। সো দ্যাটস ওয়াই দিস ইজ কল্ড multi-step, multiple mutations lead to colon cancer, genetic changes leading to tumor changes. So this is not a simple or one-step process, but always a complex and multi-step process. Initially প্রথম benign ছিল, যত এগোচ্ছে, যত নতুন নতুন টিউমার আছে, it is becoming more and more aggressive. So more and more aggressive with time. Okay, so we are done for today. Very difficult topic, but you have to face questions from this because it's a very conceptual. Please read books as soon as you get time after the fest and all. তাড়াতাড়ি পড়বি এবং পড়ে এমসিকিউ করবি. Once you do the MCQs, the close options you will select and judge, then the understanding will be more good for you. Okay, thank you.